All right, let's dive into one of the most incredible processes happening inside you right now. We're going to explore how the genetic code, the literal software of life, gets read and turned into the proteins that make you, well, you. I mean, just think about that for a second. Tucked away in almost every single one of your cells is this amazing blueprint, your DNA. But a blueprint is pretty useless if no one can read it, right? So the huge question is, how does a cell take this complex code and actually build something real from it?
Well, the answer is a process called translation. And honestly, the best way to picture it is as a molecular assembly line. The master blueprint gets copied and sent from the head office, that's the nucleus, out to the factory floor, the cytoplasm. And it's there that a whole crew of molecular machines gets to work. So let's grab our hard hats and go check out this factory in action. Okay, so first things first, let's take a look at the instructions themselves. How is this message even written?
The first thing you've got to know is that this blueprint has its own language. And the whole thing, the entire language of life, boils down to the number 3. It's read in 3-letter words. Each one of these words is called a codon, and it's basically an instruction for which building block, which amino acid, to add next. It's such a simple, elegant system, and it writes the instructions for everything alive.
So we've got the language down, but a blueprint is no good without the crew and the machinery to actually read it and build from it. So let's meet the key players that make all of this happen.
First up, we have messenger RNA, or just mRNA. This is our blueprint. It's a temporary, mobile copy of a gene that can safely leave the protected nucleus and travel out into the busy cytoplasm where all the action is. Next, we have the factory itself, the ribosome. This thing is amazing. It's a 2-part machine that literally clamps on to the mRNA blueprint and acts as the workbench, chugging along word by word and directing the whole construction project. And finally, we have the transfer RNA, or tRNA. Think of these as our delivery trucks. Each tRNA is a specialist. One end is designed to read a very specific 3-letter codon on the blueprint, and the other end carries the exact amino acid that codon is calling for. They're the crucial translators between the language of genes and the language of proteins.
Okay, so our crew's here, the factory's ready to go, but where do they start? You can't just start building anywhere. That's where the first stage, called initiation, comes in. You know, it's funny. Every single protein blueprint, for every protein, starts with the exact same instruction, the start codon, AUG. It's the universal green light. It doesn't just say, start building here. It also calls for the very first amino acid in the chain, one called methionine. So here's how it all kicks off. First, the small part of our ribosome factory latches onto the mRNA blueprint. Then it just slides along, kind of scanning, until it finds that AUG start signal. Bam! Once it finds it, the first tRNA delivery truck carrying methionine docks in. And then the large part of the ribosome locks on top of the whole thing. The factory is officially open for business. All right, the factory is online. Now it's time to build. This next stage is called elongation, and this is where the real construction happens. It's this beautiful, rhythmic cycle. Inside our ribosome factory, there are basically 2 main workstations. You've got the P site, which is easy to remember because it holds the growing peptide chain. And you've got the A site, which is the landing pad that accepts the next amino acid delivery. This 2-site setup is the key of the whole shebang. And here's that cycle in action. Step 1, a new tRNA delivery truck arrives at the open A site. Step 2, the factory links the protein chain from the P site over to the new amino acid. Step 3, the now empty tRNA gets kicked out of the P site. And step 4, the whole ribosome shuffles down exactly 1 codon, 3 bases on the blueprint. And get this isn't happening in slow motion. This factory is adding about 20 amino acids to the chain per second. It's unbelievably fast. So this chain just keeps growing and growing, adding link after link. But how does the factory know when the protein is done? Well, that's the final stage, called termination. It's actually pretty simple. Built right into the mRNA blueprint are specific 3-letter words that don't call for an amino acid at all. They're just a red light. Their only job is to signal, stop, end of the line. So what happens when the ribosome hits one of these stop signals? Well, that stop codon slides into the A site, and the whole operation grinds to a halt because there's no tRNA that can bind to it. That's the trigger. The machinery cuts the brand new protein chain loose, setting it free in the cell, and just like that, the whole factory falls apart, ready to find a new blueprint and start all over again.
We've been deep in the weeds of the mechanics, but let's just pull back for a second. Why is this microscopic factory so incredibly important?
This quote really just nails the elegance of it all. You're decoding a simple, one-dimensional code to create a specific, functional, three-dimensional machine. It's the exact moment that pure information becomes physical action.
And this is why it matters so much. Every single thing your body does, thinking, moving, digesting your food, fighting off a cold, all of it depends on proteins. And every last one of those proteins was built by this exact process, happening trillions of times a second in nearly every single one of your cells. It's staggering.
The precision is just, it's breathtaking, right? But it all hinges on one thing, a perfect blueprint. So it leaves you with a really big question. What happens when there's a mistake? A typo in the genetic code? What happens when the factory gets the wrong instructions? That's a thought to chew on.